Okay, I'm going to talk about some of our work for using a new type of computational miniature microscope for large field fluorescent imaging, in particular for neuroimaging, in a wearable miniaturized form. To start, let me give you a brief overview of my group as there's a brief introduction. I lead the Boston University Computational Imaging System Lab. In general, we do three things that sort of put everything together into the final imaging device. So we do up to hardware designs and modelings and algorithms, and toward various different scientific biomedical applications. So towards that, there are three things we primarily work on. One is to modeling and computational imaging in scattering medium, which we will see some of the components will be important for the understanding of this type of computational microscopy for neuroimaging. The other aspect that we do a lot is computational imaging with deep learning. In particular, we focus on how to build reliable and generalizable machine learning tools for biomedical analysis. And finally, we do a lot of neurophotonics, benchmarking, leveraging the resources recently established in my institutions. And one of the projects I will show you today is for wearable neuroimaging application. So why do we do this? So let me give you a little bit of broad perspective, but then I dive into the techniques we are building. Generally speaking, we're interested, as I mentioned several times, wearable microscopes. In particular, if you look into the fields, there have already been a lot of interesting work being done in this field, at least in the past 20 years. So seminal work is known as miniaturized microscope, or these days with a term called miniscope. So the problem with miniscope is that it can only provide a very small field of view because it's built on a grain lens acting as objective lens. So intrinsically, the field of view is limited by this objective lens. So as this kind of microscope being widely adopted and used in many, many different labs across the world, it has been soon realized that the field of view becomes the key limitation. So the ultimate goal for this project for our microscope, Computational Miniature Mesoscope, with the acronym CM², the goal is to build a microscope that have dramatically larger field of view, 2 orders of magnitude larger compared to the miniscopes, such that the field of view matches the size of the neocortex of a mouse. So on the right, you can see an illustration that sort of gives you an idea of the field of view we aim to achieve in this context. So the other aspect I just want to mention that it's not just you need to enlarge the field of view, but you also need to appreciate that this is intrinsically a 3D problem. At least you need depth sensitivity in order to fully accommodate for the full cortical surface for neuroimaging, is because intrinsically the surface of the cortex is curved. So this is a real data taken from some other groups, where on the bottom in figure B shows you the curvature of the cortical surface, and it spans approximately 1 millimeter in depth. So in other words, if we wanted to truly achieve this 1 centimeter square field of views, the depth sensitivity needs also accommodate 1 millimeter depth of field in that range. Okay? So indeed, this has been one of the key developments in tabletop systems. So I will review 2 recent work just to give you some context. So one system is known as with the acronym of RUSH. So the idea is that you can put several different lenses. So you sort of have this macroscopic wide field of view, live field-ish system, except it's very bulky. But the benefit of doing this is that by orient the microlenses towards the correct orientations, you can correct for the surface curvatures of the cortex, as I just mentioned, and stick everything together to form a wide field of view image and achieve single cell resolution. So then earlier this year, there's a simpler version of a tabletop system to achieve similar goal known as Cosmos. So what they do is, again, inspired by light fields, but the simplification is they use two lenses. Okay, so they use two half lenses cemented together. Each of them give a slightly different depths. So based on this, you can, and also it's a macro lens system. So based on this, you can see both wide field of view and single cell resolution. Okay, so based on this, hopefully you get the big picture of the motivation of this work. So the vision, which we're not there yet, but the overall vision is we wanted to build a system similar to what I just revealed, but with a dramatically smaller package. Okay? So for wearable applications, to give you some context, to wear on a normal mouse, so the weight needs to be less than 5 grams. Less than 4 grams will be even better. 5 grams is doable. The size needs to be, or the footprint of the whole thing needs to be around 2 by 2 by 1 centimeter cube. Okay. So I'm going to talk about today is the first generation of this technology. So I call it Gen 1 CM square. So how we do this is in the imaging side, we use a 3 by 3 microlens array. And just like in every light field microscopy technique, once we capture the raw data, we need to perform a deconvolution to recover the 3D distribution, which I will talk about more in detail later. The other aspect we spend a lot of time on is to engineer the excitation illumination, because for wearable device, you wanted to capture in vivo data, you need to have a built-in illumination to integrate it with your imaging system, which I will also touch upon later. At the end, in this Gen 1, the field of view we achieve is around 8 by 7 millimeter square. The depth of field is much longer, 2.5 millimeter. Resolution is around single cell resolution. So lateral is 7 micron lateral resolution. So you can resolve single neurons, but now with very high details in the subcellular region. The axial resolution is worse, 150 microns. And the working distance, which is also important metric for how you can place the device on top of the cortex, the mouse, is around 10 millimeters. So that's by design. All right, so a little bit more breakdown of this device. So as I mentioned, there are two components: the imaging component and the illumination component. So let me talk about the illumination first, since it's relatively easier to explain. So what we do is, because of sort of the entire footprint or how large the device can be, we cannot use a traditional design. So what we came up with is this LED array, or 4 LEDs placed around the micro lenses, and so they obliquely illuminate the scene we want to image. And we optimize their distribution, as well as some 3D printed apertures to sort of shaping the overall illumination region by Zmax. Okay. Once we do that, we fabricate everything and put everything together. And you can see the experimental results match quite well with the simulation. So some of the challenges. So of course, in most of all the microscopes, including the miniscopes, the illumination is built on epi-illumination. So whereas what we use is, as I mentioned, off-axis illumination. So this creates some challenges. So in particular, the reason is because in fluorescent microscopes, we need to use excitation and emission filters. And these high efficiency filters are typically built with interference effect. So this is the particular curves I show here is the filter we use in our experiment. So based on the data sheet we got, you can see that as you change different incident angles, the emission filter have a blue shift of the passband and also have varying different transmittance. So this is something that we need to overcome, this sort of spectral leakage problem. So the first version we built this device, the solution is quite simple. So we just use cross polarizers placed between the LEDs and the imaging path. And we're building this Gen 2, and hopefully we can come up with better ideas. Anyway, so that's the illumination path. So for the imaging path, so we use a 3x3 Microlens Array directly placed on top of the sensor. So these pictures I show you here are CAD models to scale to the main device. So to give you an idea of how everything are put together. So a little bit background. So Kevin already gave a nice talk talking about the differences of different light field configurations. So I'm very brief. So basically, light field microscopy, when it started, what people do is to place Microlens Array at the imaging plane of a microscope. So the data looks like something like a lot of different circles, and within each circles, you see different angular informations, whereas different microlenses sample the positions. So the problem with this is that it suffers from strong background fluorescence and scattering. Although this has been used in the miniature LFM, so they also call it mini LFM, a miniature light field microscopy for wearable applications. It is clear that when we studied this will place some limitations for other applications. So what we like to use is the so-called these days called Fourier LFM configuration, where we directly capture different views from different microlenses. So some nice work have done, including our chairman in this seminar work. So just by looking at the raw data, you can see the similarity between the raw data taken from typical Fourier LFM setup compared to our CM square setup. So the key difference is that because we're placing the microlens directly in front of the microlens, in front of the sensor. So we don't have a telecentric system to confine the potential overlap from different views. So in fact, the majority of our imaging data have at least 40% or 50% of overlap between different views. So this is necessary if you wanted to look at using this setup to look at a wide field data. If you look at very narrow small field of views, of course, the overlap will go away. Okay? That's one aspect to overcome in reconstruction. The other aspect, as I mentioned, is when you look at biological samples like a brain, you need to overcome strong background fluorescence and tissue scattering. And I will show you some experimental data and how we overcome and how robust the CM square setup is to these two issues. Okay, so a little bit more details on how this setup works. So this is a depiction of how the PSF works and how it encodes 3D information. So if you have a fluorophores closer to the Microlens Array, so you're gonna have a 3x3. So in this 2D cross sections, you see 3 dots in your, in the drawings of the PSF. So as you move away, as the fluorophore move away, you see that the separation of the 3 foci gets closer. So basically, it's a kind of disparity gives you the depth information, just like in every other light field technique. Okay, so this is a 2D view of this, how this array PSF look like in the device. These are real experiment data. So if you put everything together and perform a maximum intensive projection, so this is a PSF, 3D PSF of CM square. And on the top is the XY view, and on the bottom is XZ view. So in particular, we perform this MTF analysis just to give you a little bit clearer picture of how to quantify the resolutions. So in particular, if you look at the shearing of the 2 side foci, and relate that with the corresponding Fourier coverage in the 3D MTF, so you can draw a clear analogy between how this device works and tomography. So each of these shared foci can be think about as a projection along the particular direction, and correspondingly, if you recall how the Fourier slice theorem works, that's the reason where you have this tilted band in the Fourier space in the MTF analysis. Okay? So together you can use this this analysis to understand what is a theoretical achievable axial and lateral resolution based on the coverage in the MTF space. Okay, so putting everything together, just to sum up how CM2 works. So we have some fluorescent object. We shine this with our built-in illumination. We capture some images, something like that, with particle clouds, using the pre-calibrated PSF and perform 3D reconstructions. Okay, then we'll achieve volumetric reconstruction. So very briefly, so the reconstruction works by solving regularized least square problems. And we solve this with an algorithm called ADMM. And the regularizers we have tried in this work is a combination of L1 norm, which enforces direct sparsity in the 3D space, plus a 3D total variation. So that helps. So total variation and L1 together helps to suppress a lot of the unwanted artifacts from scattering and background fluorescence. All right, so a few experiments, various experiments focusing on different aspects. The first one is on the tilted resolution target, just to give you an idea of the lateral and axial sectioning. So this is a reconstruction, and in the fly through in the movie, you can see sort of the sectioning capability we can get from this single shot measurement. The resolution we got is around 7 microns. And if you look at the axial MIP cross sections, you can see the tilting angles of the target. Okay. So next experiments we look into are these particle clouds. So these are 10 micron fluorescent particles. And we compared the 3D reconstruction with standard Widefield fluorescent microscope with 0.25 NA objective. As you can see that the lateral positions are matched well, but the axial section capability clearly is worse than 0.25 NA. Okay, so this is a little bit more quantitative analysis on axial elongations. So at the end of the day, so after the reconstruction, so the elongation is somewhere between 0.1 NA and 0.25 NA. So let's give you sort of intuitive metric of how well this axial sectioning can be. So the next test we did is to go ahead to look at a 3D sample, but is spread over a 3D curved surface. So this aims to emulate the cortical surface, the curved cortical surface. So we 3D print this mouse model, but it's in a clear volume. Okay? So then on top of the surface, we spread some fluorescent fibers. Okay? So this is what you see in the measurement raw data. And after performing reconstruction, perform a depth-coded max intensity projection, this is what you see. And some fly-throughs of this reconstruction result. Okay. So again, to benchmark the results, we compare the reconstruction with a 10x 0.25 NA typical Widefield microscope. So a few things you can notice. One is that when we perform the benchmark experiments in the typical Widefield microscope, because we are doing manual focus, which means that even if when the objects are out of focus, as long as you have strong fluorescence, they will appear as a blurring blob, right? Appearing in the measurement. Whereas in our case, because we're performing a 3D deconvolution, we're trying to only recover the features that are in focus, and the algorithm will reject all the out of focus informations by the virtue of this reconstruction process. So this is clearly demonstrated by these field view, zoomed in views. So you can see that the in-focus features are matched relatively well to the benchmark, but a lot of these sort of clearly out of focus features are rejected by the reconstruction. So last but not least, let me just focus on a little bit more on the issue of scattering. So so basically, we as I mentioned, when we wanted to apply the device like this to bring imaging applications and or in general apply to an environment with strong tissue scattering, we have to understand how well it will behave when the device is imaging towards a scattering environment. So how we did this is to build a series of phantoms. In particular, we have 8 phantoms. So the phantoms are embedded with 2 kinds of particles. One is 1 micron polystyrene microspheres. They are so these kind of spheres does not have fluorescent dyes, so they are acting as scatters to blur your measurement. So the second type of particles is 1 micron green fluorescent particles. So these are actually acting as background fluorescence. And because the size is much smaller, so 1 micron, much smaller than the resolution we can achieve, so they can only be acting like the background fluctuations. Then finally, we have the targets. So in this particular case, we are imaging 25 micron fluorescent particles. So these are the larger particles. So these are the actual targets we want to image. We further calibrated the mean scattering frame path. So as noted in each of the pictures, so you can see it gives you some ideas of the corresponding scattering strengths. And overall, the samples with an anisotropy factor G equals to 0.9, so greater than 0.9. So it's similar to tissue, but it's a little bit larger than what we want. So these are the raw zoom in measurement from CM squared. So to give an idea, so you can see that the contrast in general goes smaller. And also the seeding density, I should emphasize, the seeding density of the targets, these are the bright spots you can see directly in the raw measurement. We control as the same, but because of the stronger scattering, you can see clearly in the raw measurements, you can see fewer and fewer particles. This is because a lot of the particles in the deeper layers, they become so, the signal becomes diminished because of scattering process. All right, so because of this, we run reconstructions. So we have some background compensation algorithm, which I won't have time to talk about. So you can refer to that in our paper. But anyway, more or less the same algorithm, plus some background compensation process. We run the reconstruction. In general, this is a trend you observe. If you have a very clear volume, you can recover the entire volume. But as the volume becomes more and more scattering, you have less and less depth imaging capabilities as you expected. So we quantify this. So how we quantify this are 3 aspects. One is just look at the raw measurements and quantify the signal to background ratio. So you can see that it's generally decreasing from 70% contrast to 20%. And 2 other aspects, one is scattering mean free path, so I already mentioned. The other is we measure what's the reconstruction range you can recover by basically visually measure the extent of this reconstruction volume. And you can see that in general, when the scattering mean free path is shorter than the native axial resolution, and also the native axial elongation of 25 micron particles, will now be able to resolve particles at different depths, which is around sample 6. All right, so the one last sample we tried is to put scattering particles, so the same phantom we used in the previous experiment, but now in a curved geometry. So the point here is that even under scattering, we want to show that we can still utilize CM squared's 3D imaging capability to compensate for the curved surface. Okay, so indeed, this is what we achieved. So as the right-hand side is a repeated flying through of the 3D reconstructions. On the left-hand side, we zoom onto this particular regions under two different magnifications, and one with 0.25 NA, so you can see a little bit more particles, and a little bit more zoom in 0.4 NAs, so you can see much better with better sectioning. So you can see one-to-one correspondence. So you can see that the reconstruction can still pick up the in-focus features very well. All right, so a little bit just towards the future work. So as I mentioned at the very beginning, the motivation of this work is towards brain imaging. So we're still working on that. So at the time we built this version 1 device, this is what we can achieve. So these are fixed brain slices, and we run the same imaging and reconstructions, and this is kind of the reconstruction we can achieve. So the main limitation we found is that as I mentioned, the excitation is oblique. So overall, the device suffered from low efficiencies. So this sort of hampered the overall imaging quality we can get from this kind of biological slices. We're building the second version, and hopefully we will be able to report better results next time you see this paper. So with that, let me briefly summarize. So CM squared, to this project, we want to achieve centimeter square field of view, millimeter square depth of field. In the Gen 1 version, we kind of achieve these two first metrics. So the lateral resolution, we ultimately want to get to sub 5 micron lateral resolution, so we can clearly resolve subcellular informations. Now we are at 7 micron lateral resolution, 150 axial resolution, which we also want to improve. The important aspect is to make sure the device is robust to bulk scattering and characterize how it can use in tissue environments. And eventually we're going to use this for head-mounted in vivo brain-wide imaging applications. And with that, let me acknowledge my students in my group. In particular, Yujia is the one that did pretty much all the work, and our collaborators in Boston University, in particular in Neurophotonics Center, and funding from NIH. And I'll be happy to take questions.
Okay, thank you very much, Lei Tian. I don't know why, but my video is blocked by the technical support. ¿Hay alguien de INT por ahí? Tengo el vídeo bloqueado.
Dígame, doctor.
No sé por qué.
Dígame.
Sí, tengo el vídeo bloqueado. No sé por qué. Ahora. It's okay. It's okay. Okay. Thank you very much.
I can see you now.
Sorry for going to Spanish. You know, when we have problems, it's much faster. I had my video blocked at the beginning of your talk, and then I couldn't make your presentation. So really, my apologize. So I can make it now. So this is Professor Lei Tian. He's the head of the Computational Imaging Laboratory at the Boston University. And okay, as in very often today, apart from me, that I'm an old professor, we are collecting a collection of very bright and young researchers. So thank you very much for being here, Lei. I have some questions about your system while waiting for the questions in the chat. So your miniscope, then you are not using microscope objective. You have directly the Microlens Array in front of the object. That's correct?
Yeah, that's correct. That's the main challenge. That's the main challenge.
Main challenge. And how do you do? Because have you okay? This Microlens Array is manufactured for you, especially with some special requirement for aberration, curvature, or something like that, or it is off the shelf Microlens Array?
So it's off the shelf Microlens Array. So in fact, you can buy this very cheap. So one of the things I forgot to mention, thanks for the question. So I forgot to mention is that to emphasize is, so at the very beginning I mentioned for a wearable device, the whole weight needs to be 4.5 gram, less than 4.5 gram.
Wow.
Because of that, the optics needs to be very, very light. So this Microlens Array we use is a plastic molded.
Wow.
Plastic molded microlens. So it's not very good quality. In fact, if you look at some of the zoom in PSF we give in the supplementary, it's very strongly aberration, have a very strong aberrations. So all of this sort of are partially compensated by the algorithm. So
you have demonstrated that with an array, low quality and low price array of microlenses, you can obtain better quality than with a microscope objective, for example, of numerical aperture 0.25. That's correct?
No. So it's around point. It's around point. I would say around 0.15 overall, which is kind of reasonable for this kind of application, because I mentioned we only ultimately want to achieve 5-micron resolution. So you don't really need a very high NA system. So overall, generally what we found is the image quality after deconvolution is comparable to a 0.1 or 0.15 objective lens.
But it means that the convolution procedures applied to light field images are more effective than the convolution procedures applied to conventional imaging? Or you compare your case with the convolution with the other case without the convolution?
Right. So it's hard to say, because so in the benchmark we showed, right? So you're right that we didn't do a deconvolution. So these are the raw data from fluorescent microscope, standard tabletop system. They don't have much aberration. Whereas in our case, if you look at the raw data, you can kind of see the aberrations. But after doing deconvolution, in particular, the key thing is the deconvolution involved adding priors, L1 and TV. Those have a strong effect, and in particular are effective for the kind of samples I showed.
How do you make to avoid the overlapping between elemental images?
They have overlap. So this is one of the
you don't put physical barriers between microlenses or nothing. You put nothing between Microlens Array and the sensor. No physical barriers. Right.
So so there's no physical barriers. So directly the microlens is attached in front of the sensor. Well, not attached. I mean, we have a 3D printed holder to hold it to the correct distance. But anyway, so most of the data I show have significant overlap. So you have about 40 to 50% area overlap between neighboring views.
Yeah.
That's one of the key things. Yeah. So that's one of the key things that deconvolution needs to perform. So this is one of the reasons you would not be able to perform sort of a traditional sort of view-based backpropagation in the digital refocusing in light field, because you cannot separate the views in general.
I never had thought in this way. So you can take profit from overlapping area thanks to the deconvolution procedure.
That's right. Yeah, that's one of the key new thing, I would say, from a light field perspective.
I never thought in that way. It opened my mind. Thank you very much.
I would say there's a trade-off. There's a trade-off that we also mentioned. I didn't have time to go into it, but we mentioned in the main paper when we explained the scattering results I mentioned. Because of the overlap, you get worse contrast in your scattering experiment.
Okay, I have a quick question from Julian Colombelli. I will read it for you. Mm-hmm. Nice talk, of course. You mentioned a working distance of 1 centimeter. Is there a way to easily shift or modulate this distance axially to repeat volume measurement closer or farther without moving the camera chip position, and to extend the overall imaging volume even if at the cost of several imaging iterations? Even more, any electronic component and electro-optical component that could be inserted to do this? So
yeah, very good question. Very good question. So a lot of these are sort of underway in the Gen 2. So this sort of broadly, if I understand correctly, this has to do with a focus adjustment. Right? So the keyword is focus adjustment. So it's a very good question. So because we are using a lens, as everybody knows, there's a nominal focus plane, right? Because the object is conjugate this far away from the lens. So the 10 millimeter I quoted with the working distance is a distance from the nominal focus plane to the microlens. This is typically needed because when you place the device on top of the skull of the mouse, you need some fixtures, and typically we have hub bars and stuff like that. Actually, in our recent in vivo experiments, we found this is necessary. Sometimes even taller is needed, like 10 to 15 millimeter is a good number. On the other hand, focus adjustment is needed, just because we're not literally needs to look at the surface. Actually, you need to look a little bit deeper inside the brain, because the top surface actually does not have any fluorescent signal. The fluorescent signal when you inject, let's say GCAM6, is actually a few microns underneath. So all in all, I guess my point is, as the audience asked, do we need focus adjustment? Yes, we need. So that's something we're trying to work on with some mechanical designs in the 3D printed housing. Up to electronic adjustment like liquid lens or stuff like that. Yeah, those are sort of a dream if we can afford the weight. Right now, it doesn't seem to be achievable because we are pretty tight on the weight. So we wanted to. So the Gen 2 we got to 4.5 gram, but it's pretty tight. Yeah, pretty tight. This Gen 1 is much higher, did not meet the 5 gram goal. So we had to cut down a lot of weight further.
Okay, Lei, thank you very much. I think we have this meet the speaker in maybe half an hour or something like that. So thank you very much, and see you in a few minutes.
Yeah, see you in a minute.
Bye-bye.
Thank you, Manuel. Bye.